Hello everyone, I'm Suman Yadav. In this video, we will talk about how adaptive immunity works. Okay, so basically, adaptive immunity is nothing but your acquired immunity. Okay, it is one and the same only, acquired immunity or adaptive immunity. So, as we know, immunity is nothing but the defense system of our body, how it control the infection from the pathogen. Okay, so there are two kind of immunity, just a basic idea that we have a innate as well as acquired immunity. Okay, so how it is different from the innate immunity basically. So innate immunity will discuss in the another video. So here we will focus on acquired immunity. But what is the basic difference that we have to understand? Innate immunity is the immunity which is present since birth. So we are born with innate immunity. So it is present since birth. And this acquired immunity is acquired after birth, and it is get activated only on exposure of antigen, only on exposure of pathogen. So acquired immunity activated only there is when pathogen enters. Let's say a person is when infected with the chicken pox, when it is suffering from chickenpox. So basically, acquired immunity against the chickenpox virus get activated. So it is nothing but the acquired immunity always work only, get activated only when there is a exposure of the specific pathogen. So second difference is acquired immunity is very specific. It is antigen specific. Antigen means nothing but the protein or your protein present on the cell membrane. It is specific for the all kind of cell. For example, human cell have different antigens. That is the surface antigen present on the cell membrane. Then your bacteria has different, virus has different. So this acquired immunity is specific for the pathogen, or you can say it is very specific for the antigen. But innate immunity is not that much specific. Basically, it can identify the uh, outside pathogen, that's self and non-self, but it is non-specific. Basically, it will act same for all kind of pathogen. So it is not specific, but our acquired immunity is specific for the specific antigen. Let's say for chickenpox virus, we have a activated active acquired immunity. This acquired immunity will not work for the another virus like hepatitis B virus or C virus. So it is very specific. Also, the most important property which we use in your vaccination is nothing but the memory. So, acquired immunity stores the memory. What does it mean? It means that once we are infected with the certain pathogen and our acquired immunity get activated, it will remain stored. So when there will be the difference in the primary and secondary response. So once the pathogen encounter for the first time, when pathogen enter for the first time, what will happen? Our acquired immunity against that pathogen get activated. So the response will be for the first time, response will be the slower one. So this is called as primary response. So primary response will be of course slower, but it will, when the acquired immunity activated, it will store its memory. So certain cells which are involved will discuss now. So that will remain its memory, that will that cells, activated cells will remain as a memory, and they will activate when there will be a second time pathogen is getting enter. So that response is called as secondary response. So of course there is the difference in the primary and secondary response. The primary response is slower because our acquired immunity take time to get activated against that pathogen. But second time when the pathogen enter, our immunity has the storage, it has a memory for that. So this is more intensified response, more incentivized response. So that's why this method basically this property is used for the vaccination purpose. What we do, we give the dummy pathogen, or you can say antigen part of the pathogen, or inactivated pathogen. And in vaccine, basically, what is it? It is inactivated pathogen. Okay, and so once it enters the against that antigen, what will happen? Our primary response is of course slower, but it will not affect our body. But our body get prepared for that pathogen. If you are getting a poliovirus vaccination, so our body get prepared for that poliovirus. It will feel that there is a infection of poliovirus, and it will activate the our adaptive immunity, and that immunity will get stored. So once when there is actually entrance of the primary your poliovirus, then body will act, give its fast response, intensified response, so we do not get ill. So this is the way of your working of your acquired immunity. So let us discuss what are the cells involved in acquired immunity and how it works. Okay, so cells involved in acquired immunity. Cells involved in acquired immunity. So there are only two cells which is involved in acquired immunity, because as it is totally different from our innate immunity, there is number of cell and number of methods for the innate immunity to work. But here the acquired immunity is very precise, very specific. There are only two kind of WBCs which are working here. That is a type of lymphocyte, type of WBC only. Okay, so in WBC we have a granulocyte and agranulocyte. So the type of agranulocyte, that is lymphocyte, we that is involved in acquired immunity. So we have two kind of acquired immunity: B lymphocyte, two kind of cells, that is B lymphocytes and T lymphocytes. So both are lymphocytes only, a type of WBCs. So as we know, the lymphocytes are produced in, or even we can say all blood cells are produced in bone marrow, right? Let's say this is bone. So here, somewhere here, there is presence of stem cell, that is red bone marrow. So here all bloods are produced, right? So all blood cells are produced. So B lymphocytes and T lymphocytes both are produced in, where it is produced in red bone marrow, but maturation is different in both cases. That's why the name maturation is different. B lymphocyte is produced and matured in red bone marrow only, but the T lymphocyte is produced in red bone marrow, but it is mature in the thymus. So thymus is the primary your lymphoid organ. So it is mature, it get matured and differentiated in your thymus organ. So that's why the name is T lymphocytes and B lymphocytes. Now, the both of this work differently. There are two cells involved, but these two cells work differently, and they uh, fight against the pathogen. So B lymphocyte give the humoral immunity. Okay, and T lymphocyte give cell mediated immunity. So their way of working is different. So that's why the name is cell mediated immunity and humoral immunity. So what does it mean? What is the meaning of humoral immunity? Humoral means the thing which is secreted by B lymphocyte is a protein, a kind of protein, that is antibodies, which is very specific for the antigen, and it is released in the body fluid. So humor means body fluid basically. So body fluid means blood or lymph. So the B cells, let's say this is B cells, B cell release its antibodies. This is antibodies into the fluid, body fluid. Let's say this is body fluid, this is blood vessel, here there will be body fluid. So it will release its protein, that is specific protein to the specific antigen, that is antibodies inside the body fluid, and this will bind to the antigen and opsonize it. and etc. So this way of working is called as humoral immunity. So this will provide humoral immunity. What about cell mediated immunity? In this case, the T, basically T lymphocyte is involved. So this T cell, when get activated, it will activate the other cell. So it will release certain chemical that is known as cytokines. T lymphocyte, especially we will see the types of T lymphocytes. So it will release cytokines, which will activate the B cell and other phagocytic cell, so that the immune system, our acquired immunity will work. So they are activating the cell directly. So that's why the name is cell mediated immunity. And here humoral immunity means it will release antibodies inside the bloodstream, and that's why the name is humoral immunity. So the B lymphocytes, when get activated, the B cell when active, it is in present in naive form or inactivated state. So when it get activated, it will let's say pathogen enter, and T cell will see the proper how method, how I mean the process, how our T cell activate the B cell. Okay, so we'll see in few minutes. So when it get signal from the T cell, B cell will get activated. So it will start proliferating and differentiation. Then it will form the plasma B cell. So basically, the effector B cell. This is not effector cell. This is just a naive form of the B cell, which is specific for the specific antigen. So when it get activated, it will produce the effector B cell, and that effector B cell is called as plasma cell, plasma B cell or plasma cell. So now this effector cell will start secreting or producing the Y-shaped antibodies. And this is very specific for the specific antigen. Let's say if this is the pathogen, and if there is a antigen, B antigen, and C antigen, so this antibodies are so specific that if it is recognizing this antigen, then it will always recognize this antigen only. So it is very specific, and specifically it is going to bind the antigen of the pathogen. Okay, so plasma cell will produce antibodies, and so some cell of the B cell get proliferate and get differentiated into B cell to produce antibodies, but certain cell will proliferate, but it will not get differentiated. So what it will do, it will keep the memory. So it will be called as memory B cell. So as we discussed earlier, so it will form the memory also. So it will be stored for the future infection. So when there is once again entry of the pathogen, so this cell will be ready for the releasing the antibody. That time it will get differentiated and will produce the antibodies. Okay, so general structure of antibody is nothing but, let's say there are we called as H2L2, two heavy chain and two light chain. So it is nothing but the protein only, polypeptide chain only. So how it is, what is the basic structure? It is shaped structure like this. This is the two heavy chain. This is two heavy chains. Okay, they are bounded by your disulfide bond. Basically, they are bounded by disulfide bond. Now, in both, there will be two heavy chain and there will be two light chain. This is light chain. These are again bounded by your disulfide linkage. Okay, disulfide bond. So here, in case of antibodies, how it recognizes the specific antigen, there are the two reasons here. One is your very specific reason, that is your variable region. Variable region. Okay, and other part, from this part to this part, this is called as your constant region. I'm sorry, constant region. So this will be known as constant region, which is constant for all, constant region. But this variable region is actually the reason of the binding for the antigen. Basically, antigen binding site, we can say. Okay, this is the antigen binding site. So if let's say this is your bacteria and this is the antigen. So this antibody will come as plasma cell will release in the fluid. It will come and it will bind to this antigen because it can specifically recognize, as you can see, as you can compare with the key and lock will always will open with the specific key only. So in the same manner, this is nothing but uh, receptor type. It will bind to the specific protein only, specific antigen only. This antigen is nothing but protein only, protein present on the cell membrane. Okay, so this is the way of your working of your antibody. It will bind, it will opsonize, and it will attract the other phagocytic cell to eat this cell. Also, it will basically neutralize the toxin released by your pathogen, which will which can cause illness inside the body. So basically, antibodies help in bringing other cells to act on it by opsonizing the antigen, and also it neutralizes toxin. Now, what are the cell mediated immunity? This B cell get activated by your T cells only. Okay, it cannot get activated by itself. It will activate it when T lymphocyte will give signal. It will release cytokinin to get activate the particular specific B cells. Okay, so let's see how. So first of all, T lymphocyte in cell mediated immunity, we have a three kind of your T lymphocytes. So we have first type is known as helper T cell. Helper T cell. Okay. Second one is your cytotoxic T cell. Okay. And third one is your suppressor T cell. So we have a three kind of T cell present in its naive state, means inactivated state. So helper T cell, what is this? This is nothing but alarm. It will generate the alarm to activate the your acquired immunity. Basically, it will sense the pathogen by your antigen presenting cell, and it will get activated and it will release the cytokines to activate the B cell. So basically, it will activate B cells, phagocytic cell, and also the cytotoxic cell. So basically, its role is to release cytokine and to activate the other cell. About the cytotoxic cell, once it is getting activated, means these are very specific. The T cells are also very specific to the specific antigen. They carry a receptor. They carry the T cell receptor on their surface, which will recognize the antigen when it is bound to the certain antigen presenting cell. Okay, I'll come to that part. So cytotoxic cell is also antigen specific. This is also antigen specific. They are very specific. That's why we are talking that this is the specific antigens specific immunity. So what is the role of cytotoxic cell? To kill the infected cell. To kill the infected cell. So in case of infected cell, there is, of course, it is infected, so it can spread the infection to other parts, so it should be killed. So it will kill the infected cell or cancerous cell. Cancerous cell. So cancerous cell also get removed by your cytotoxic natural killer cell. So cytotoxic, how it works? So normally, let's say this is cytotoxic cell, it will release perforin. So this will this will give the signal to your normal cell. They will form pores and they will get killed or die. Now suppressor T cell is nothing but your, it is the kind of check, so that it should stop the autoimmunity. To stop autoimmunity, it means the defense system, our cells should not kill our cell. It should not recognize the own antigen. So basically, it is to check the cell. It will suppress the cells which is acting against our cell, the self cell. So it will stop autoimmunity basically. Okay, so we have a three kind of this cell. Now, how it is? This will work in chain. So basically, innate and acquired immunity both work simultaneously. Okay, so acquired immunity get input from the innate immunity. So in innate immunity, we have a different kind of cell. So one of the cell is your phagocytic cell. Phagocytic cell means we have a macrophages, we have a neutrophil, which will kill the bacteria. It will basically engulf the bacteria. Let's say this is macrophages. It will engulf the pathogen, and it will digest it, and it will present it on its surface. So we have this kind of cell is called as antigen presenting cell. Antigen presenting cell means they are presenting the antigen on surface. This is kind of giving signal to other cell that we are infected with the pathogen, we are in danger, please get activated. So how it basically work? So let's say one of the most important antigen presenting cell, that is again they are phagocytic only, but they give signal to the your T lymphocyte. So let's say this is the cell that is dendritic cell. What it will do? It will do the phagocytosis like this only. This will do the phagocytosis. This will digest it, and it will make the antigen presented on its surface on the some receptor that is called as MHC2 receptor or MHC2 molecule. It is kind of protein present on the surface. Okay, so not to worry if you're not able to understand this MHC2, you will learn this in higher standard. So antigen presenting cell like dendritic cell or your macrophages have certain protein, that is MHC2 molecule, on which it will digest this antigen, and it will keep the antigen here. So digested or processed antigen, it will present on MHC2 molecule. So this is nothing but MHC2 plus antigen complex. This is generally alarm for activation of your T helper cell. So this is recognized by your receptor of the specific, let's say this is your T lymphocyte, T helper cell. So if it carries the specific antigen, I mean specific recognition site for this antigen. So it will be the T cell receptor, which is very specific for this antigen. So this will able to recognize it, and specific to the T cell, which is specific to this antigen, will get activated. So once T helper cell get activated, the activated T helper cell, what it will do? It will activate the B cell. It will activate the cytotoxic T cell, and also it will produce the memory for the future. So T cell also produce the memory for the future. So T cytotoxic cell, what it will do, as we have discussed earlier, it will release the perforin, which will kill the your normal tissue which is infected. Infected tissue is killed. And of course, B cell will produce plasma cell. It will differentiate into plasma cell, and also it will form the memory. And this plasma cell will act for the current infection. That is, it will release antibodies which will act on the specific antigen. It will opsonize it, neutralize the toxin, and it will attract the other antigen presenting cell or phagocytic cell to eat that antigen antibody complex. So in this way our acquired immunity works. I hope this is clear to you. Okay, so that's it. Thank you.